excited to hear more updates on what's happening on the gene therapy side. And these talks are going to be complementary to that side, but not necessarily speak about where we are in the gene therapy process. So the very first speaker is Dr. Laura Baroncelli, who has been doing a lot of work on, she created one of the mouse models that's been widely used by a couple of researchers and companies, and she's collaborated with LUMOS and published quite a few papers. And Laura is also the recipient of our very first Gene Therapy Advancement Award grant. So from the conversations we've had and the information that was sharing, we felt that Dr. Laura Baroncelli embodies the spirit of collaborative research and has helped the whole group kind of move forward. Dr. Baroncelli, you can start sharing your slide and I'll let you take it away.
Okay.
Yep, we see your slides, perfect.
Okay, perfect. Okay, so good morning or good afternoon to everyone, according to your time zone. I'm very glad to give this talk to the first ever CCDS virtual meeting, and I would like to thank, first of all, Laura, Sanjita, Heidi, and the whole ASD Association for the great effort put in making memorable this event and for the possibility to share with you today my recent results. Unfortunately, the COVID emergency strongly delayed the research activity of my lab, and I'm not able to talk about the gene therapy project as I was supposed to. So I'm going to open this second day focusing your attention on cell-specific vulnerability to metabolic failure in creatine transporter deficiency. So let me start with the mouse model. 6 years ago by now, and thanks to the collaboration with the group of Cornelius Gross at the MBL, we have generated a mouse model of CTD carrying the ubiquitary deletion of the exons 5, 6, and 7 of the SLC6A gene, leading to the loss of function of the creatine transporter protein and causing a marked depletion, as you can see here, of creatine in the brain and peripheral tissue of mutant animals. So now let me introduce to you one of our knockout mice, here side by side with the wild-type littermate. During these years, we extensively characterized this model with a twofold objective. First of all, we aimed, of course, to set the translational value of the model for the development of therapeutics, but we are also interested in investigating cellular and molecular mechanisms of CDT pathogenesis with the goal to identify novel druggable targets. So in the next slides, I'm briefly going to show you that knockout mice share the clinical picture of CTD patients, including cognitive dysfunction, autistic-like behavior, and seizure. Then I guide you throughout the physiological and cellular alterations that we identified in the CTD brain that led to the hypothesis that inhibitory interneurons, and in particular parvalbuminergic cells, are vulnerable to the metabolic insult caused by creatine deficiency. But the first thing first, we have performed a wide battery of behavioral tasks, and we found that the knockout mice display a global deterioration of cognitive functions, ranging from the working memory, as assessed using the Y maze. Here you can see that the spontaneous alternation of mutant animals is significantly lower in this test with respect to wild types, to the field of declarative memory, as assessed using the object recognition task, where knockout mice fail to remember a familiar object with respect to a new one already after 1 hour after the presentation, later than the first presentation. And the Morris-Water maze, where mutants struggle to learn the position of an hidden platform and fail to remember its location in the probe trial. Second, with regard to the core ASD symptoms, we detected no differences in sociability of knockout mice in the 3-chamber social test, but stereotype behaviors in the rotarod and self-grooming tasks seem to be amplified, as previously reported for many other mouse lines with ASD-related mutations. So cognitive dysfunction, first point, and autistic-like behavior, at least at the level of stereotype movements, second point. But what about the epilepsy? What about the epileptic phenotype in these mouse models, which is a symptom you taught me with the greatest impact on the everyday life of children? So we use video EEG recordings in freely moving animals, and we found that around 30% of knockout mice, and this is probably an underestimation, showed at least one spontaneous seizure over the 24-hour of recording. And notably, these mice with the epileptic phenotype at the spontaneous level display on average one seizure every 2 hours, so quite frequent event, lasting on average about 20 seconds and presenting as clonic or tonic-clonic convulsions at the behavioral level, at least in most cases. And moreover, knockout mice display the more pronounced accessibility to kainic acid challenge. Kainic acid is an agonist of kainate receptors and is a drug commonly used in epilepsy research to induce seizure in a likely induced by a perturbation of brain circuit excitability. And this prompted us to examine more in depth spontaneous and evoked basal brain activity. For the first one, we used the baseline of EEG recordings we already performed. We segmented EEG signals in epochs of 30 seconds, and we classified the vigilance state in each epoch of the animals as active wake, passive wake, or sleep, according to the inspection of video recordings. And then the EEG signals were converted into power spectra by an FFT analysis. And here you can see the representative spectral plots comparing wild type and knockout animals for each condition tested. The color code represented different spectral band we analyzed. And what we found is that knockout mice showed significantly altered power in a wide range of EEG frequency, and more specifically, theta and alpha band were significantly decreased in the cortex of knockout mice, while gamma and beta were increased. So highlighting a transition from a physiological to a pathological state of network activity, which is similar to that previously observed in other neurodevelopmental disorders and patients in autistic spectrum. At this point, to clinically validate the results obtained in the mouse model, we also analyzed EEG data from 5 CTD patients diagnosed at the Scuola Institute of Calambrone, very close to Pisa. Here, I want to stress that EEG used for this analysis were those recorded at the time of the first diagnosis of these children, before the beginning of any medication, thus providing us an information about the natural condition of brain activity in CTD without the possible interference of any pharmacological treatments. And these are again the spectrum plot, the representative spectrum plot, and despite the low numerosity of our sample, we report a significant alteration of brain oscillation in CTD children with a higher power in delta gamma bands and the lower power in theta and alpha. And notably, the same power bands, but in particular, theta and alpha, are altered in the same direction in mice and patients, indicating that EEG alterations in the mouse model might be predictive of those in CTD patients and possibly identifying a network biomarker of CTD disorder. Then, for the study of evoked brain activity, we focused on our good friend visual cortex. In my lab, we had a lot of expertise in the study of visual cortex, so we decided to assess visually evoked responses using intrinsic optical imaging and electrophysiology. IOC imaging is a non-invasive technique measuring through the intact skull of the animals activity-dependent changes in the light reflectance of the brain tissue. It's very similar to the Bolt signals and of the functional fMRI. And the two main sources of the intrinsic signal are the transient deoxygenation of hemoglobin in the active brain areas and the subsequent changes in the cortical, in the local blood flow. So as you can see from this image, the more active cortical regions appear darker because they reflect less light than the less active areas. And surprisingly, quite surprisingly, we found that IS amplitude was significantly higher in knockout mice with the effect longitudinally present at all ages tested. Quite different is instead the situation, the picture emerging from the electrophysiology, since the VET amplitude and latency are comparable in knockout and wild types, thus suggesting that only hemodynamic responses are altered in the cortex of mutant animals due to a possible deregulation of neurovascular coupling. And with this pool of data on our hands, we evaluated the reliability of behavior, imaging, and electrophysiological recordings as biomarkers of CTD disorder. And we use for doing that random forest machine learning algorithm. But I'll make a very long story short. What we found is that, of course, the full dataset exhibits the best performance in discriminating between knockout and wild types at the single subject level, but even smaller datasets, such as behavior, imaging, and EEG by themselves have a very good accuracy in classifying the two genotypes. Here you can see the percentage of accuracy for each data set we analyzed. And moreover, and this will be very helpful later on in this presentation, the analysis of feature importance highlighted that the single variable most informative for discriminating the two genotypes is the Y maze. Okay, so let me now show you a very brief interim summary. First of all, I hope you are now convinced that the CTD mouse model has a high translational value for the evaluation of potential therapeutics. And this is true, of course, for our model, but also for the model from Matthew Skelton's lab. In this line, we found that the assessment of EEG activity and hemodynamic measurements might represent a non-invasive tool for the analysis of brain functions in CTD, and EEG actually is already part of the current clinical practice for the diagnosis and follow-up of metabolic disorders, and functional NIRS is an imaging method very similar to IOS and really applicable to children with neurodevelopmental disorders, so CTD as well. And actually, we, in collaboration with the Stella Maris Foundation, we are now developing a protocol of functional NIRS, and we already obtained the ethical committee approval for performing these measurements in CTD children. And finally, our findings could provide some hints about the cellular mechanism of CTD pathogenesis, but I think that many questions still remain open in the framework of creatine disorders, and, but also about the impact of energy metabolism on neuronal physiology. In particular, if you think that the expression of AGAT, GAMT, and SLC6A8 genes present a significant heterogeneity across the brain circuits. So we asked whether a cell-specific molecular pathology can be recognized in creatine transporter deficiency. And nowadays, cell specificity is becoming more and more evocative of optimized single cell NGS. So thanks to the precious collaboration with the group of José López Atalaya in Alicante, we used 10x single nucleus RNA-seq to generate a comprehensive map of the cell types, genes, and pathways altered in CTD mouse at the cortical level at least. And these are the numbers that describe the good quality of our dataset, and supervised clusterization identified 41 clusters with very distinct transcriptional profiles, so basically 41 different cell populations for which we analyze differentially gene expression. But first of all, integrative analysis did not reveal any compositional differences between the data sets, suggesting the take-home message here is that creatine deficiency is not related to the degeneration of a specific cell population, because all the cell population in the 41 clusters are basically equally represented in knockout and wild-type mice, but this suggests that the creatine deficiency is a more subtle reorganization of brain circuits. And this is also consistent with our previous work showing that a normal neuronal density in the cortex of knockout animals, and also with the neuroimaging studies in children with CTD highlighting only mild structural abnormalities. So what about gene expression? We performed the first analysis considering statistically significant genes with the FDR less than 5%, so basically a p-value usually accepted in literature, and classifying nuclei in 5 major cell types. So excitatory inhibitory neurons, oligodendrocytes, astrocytes, and microglia. So for the moment, we grouped together the 41 different cell populations we were able to dissect in 5 major cell populations. And while we failed to detect any regulated genes in astrocytes, and this could be consistent with the apparent lack of creatine transporter expression in these cells, you can see that excitatory inhibitory neurons, but also non-neuronal cells, exhibited gene expression changes, that suggesting that CTD pathogenesis could present a multicellular profile with a complex network of cell autonomous but also non-autonomous activities. Moreover, using gene ontology analysis to classify for functional modules, we found that genes associated with synaptic transmission and the regulation of circuit excitability were enriched for dynamic expression across the genotypes, both in neuronal and again non-neuronal cells, indicating that CTD should be regarded as a synaptopathy. And accordingly, we also found that around 25% of our list is also mapped to the single database. So at this point, we asked how the dysfunction of discrete brain populations can impact on different aspects of the CTD phenotype. And since previous data from our lab showed a specific decrease of GABAergic synapses in the cortex of knockout mice, we decided to focus on these cell populations. And in this framework, parvalbumin fast-spiking interneurons are a very good substrate to explain at the cellular level the CTD pathogenesis, since the prominent expression of SLC6A gene in these interneurons, documented very recently by the lab of Stephen McCarroll and the Allen Brain Institute, and that we also confirmed in our single cell experiment. And using immunohistochemistry, we also found that the density of parvalbuminergic synaptic puncta is decreased in the cortex of knockout animals. So based on this data, our working hypothesis is that the density of parvalbuminergic synaptic puncta is decreased in the cortex of knockout mice. And that the alteration of this cell population crucially contributes to the etiology of CTD disorder. And to corroborate our theory, we generated a conditional mouse model with a specific deletion of creatine transporter in parvalbuminergic cells. As you can remember, a few slides ago, we have seen that the Y-maze is the most informative variable for classifying knockout and wild-type mice. So we use this very simple behavioral task as a first-line screening for the conditional mouse line. And what we found is clear impairment of spontaneous alternation in parva knockout mice, suggesting that creatine depletion is sufficient for the determinism of the neurological phenotype and motivating us to test the role of these interneurons in CTD impairment of brain dynamics. And let me show you the results. First, we found a significant increase in the amplitude of IO signals in the visual cortex of parva knockout mice. Here I need to acknowledge that the delta between mutants and wild types is not as much as pronounced for the ubiquitary mice, but considering that parvalbumin cells represent a low proportion of the whole neuronal population, about the 5–10% in the cortex, these results strongly suggest that parvaneurons are one of the main drivers, in the regulation of hemodynamics in the CTD brain. And equally interesting are the results of EEG, with parva knockout mice showing a higher susceptibility to kainic acid challenge. No spontaneous epileptic seizures were found, but still higher Rasen score, lower latency to epileptiform activity, higher frequency and duration of epileptic bursts, indicating that the lack of creatine in parvalbumin cells actively increases the excitability of brain circuits and exacerbates the epileptic phenotype. So, well, this is the end of my story. Just a few take-home messages. First of all, we report the absence of major cellular deregulation and degeneration, and effects only at synaptic level. So we think that creatine deficiency most likely is a disease of synapses. Second, we found that parvalbumin cells are vulnerable to metabolic alterations, and that creatine depletion may affect parvalbumin postnatal maturation, thus impairing cognitive processes and excitability of neuronal networks. And finally, we hope that this knowledge will allow us to identify novel potential targets for pharmacological treatment, since drugs targeting subtle circuit defects have already been shown beneficial effects at the preclinical level, at least, in other neurodevelopmental disorders such as Fragile X, Down, and Rett syndrome. Okay, this is the list of the people which are part of the creating group. I would like to thank in particular Raffaele Mazziotti, Francesco Cacciante, and Giulia Sagona for the work in the animal model, and Roberta Battini and Annarita Ferrari for their essential contribution in the study of patients. And of course, the group of José López Atalaya for the transcriptomic experiments. Let me also mention the funding agency and companies which made the research possible. And last but not least, I would like to express my warm gratitude to you guys, to the Parent Association, Extraordinaire, and ACD for their fundamental support of my research. And thanks to all of you for